in the space for class just because I want to, first of all, I don't want to have everyone on pins and needles until the end of class, although I technically did already know what grade you earned because they were on Sakai, but also because passing out exams takes time. So, and I want to maximize our in-class fun time. Fun. Of course, I think it's fun. Hopefully you think it's fun too. Okay. All right. So first things first, I'm going to talk about some housekeeping stuff about exam 2 for those of you who are curious. So the average for exam 2 was an 82.6. So again, last exam I think the average was 88, which is a little high. Of course, I like to think that's because you all are wonderful and intelligent and excellent problem solvers, which of course is true. But the average for exam 2 was lower. It's closer to what I would expect for an average normally. But it's not unusual for the averages to fall or to go down a little bit with a passing exam because the material gets more complex as the semester progresses. Um, it gets more complex, and the volume of material also with the chapters can get a little heavier with subsequent exams. The exam score distribution looks like this. So we had a lot of students scoring. So again, if you scored above an 80, great. If you did and you're not happy with it, I mean, you can always come talk to me. I'm happy to talk about exams, about study strategies. I would reiterate at this time, if you weren't happy with your grade, I was going to say I'm more than happy to talk to you about study strategies, but the one blanket study strategy that I think could apply to everyone is consider working with your friends in class. The people at your table, people maybe you know from your major or from an extracurricular or something. Start studying side by side, work together in groups, even just once or twice in an exam session, working with other people. They can help you see a different perspective. And I found as an undergraduate, I always of course, I studied with the same 3 people the whole time. But that doesn't work or apply to everyone. But I highly recommend, branching out, talking to people, and forming study groups. That's really the best and most efficient way. If you look at your exam and you see a big slash through problem 10, which you should, because everyone should have a slash, don't be alarmed. There was a mistake on problem 10 that I really couldn't correct with words, so I threw it out. So on your score, you'll see your score out of 98, and then adjusted to be out of 100, because each exam, the 3 exams are worth 100 points. On the ATCA's problem, there was a little bit of confusion there. Technically, you aren't on the hook for knowing whether or not, I think it was BTP is a purine or a pyrimidine, as long as you made an answer that went with committed to either being a purine or a pyrimidine, and you were consistent in your answer and and included certain criteria in your answer that are detailed in the key, you got credit. So I knew I messed up on this problem, and so I tried to be more, as long as I could see you were following the right thought process, you got at least half credit. Most people, if it was consistent, you got full credit. You also passed back. I also passed back the dope sick assignments. Overall, I was super pleased with the assignments. I could tell everyone read. Everyone was at least a little bit interested in the book, and everyone made an 8 or above. So extra points were awarded for if you effectively tied in a personal experience, if applicable. Not everyone has a personal experience. That's okay. You're not going to be penalized if you don't. I really looked for if you introduced a perspective that was not discussed in the book. So several of you tried to analyze the motives, provide doctors. I really like that. Some of you tried to pin down what you thought were the biggest contributions to the crisis, and I really um, like taking the material from the book and putting your own spin on it rather than summarizing the book was okay. You still made a good grade if I thought in your mind if you did a good job talking about the book. But what pushed people over was telling a really good story and tying it in and adding your own opinion and ultimately taking the evidence in the book and making your own argument or the idea of showing something rather and illustrating a concept through a little story rather than telling you what you think. And in the next couple of weeks, I will, if you looked at this and you're kind of like, I should have gotten better, or I'm working on writing my own essay that will be a response to this, and I'll post it in the next couple of weeks. That could perhaps help you as you're writing your next essay if you weren't impressed. As always, I'm more than happy to talk to you about it. And then one more not immediate class related thing I'd like to talk about is that there's going to be an extra credit assignment. And this extra credit assignment is 5 points on your exam grade. Yes, doing this extra credit assignment it did make the difference between some people's grade last term. So really think hard about doing this. Um, my motivation for this assignment, and I have posted the information on Sakai, is because at the beginning of the semester we went and we walked through Bloom's taxonomy. And Bloom's taxonomy, where it's the pyramid of learning skills. At the base, we had understanding, and these are the skills that basically where you memorize things. And you've taken half of biochemistry, and everybody here knows that memorizing is super important for biochemistry because you have to have that base level of memorization to build on top of. But if you've gone through, you've worked your problems in class, you've taken my exams, you know that the higher levels of learning, so analyzing, predicting, those are also very important at applying the things that you memorize to new situations. But at the absolute top of the pyramid is creating. And I think it's really important, and I think that a lot of this, I believe that the skills that are used in creating something like art or an independent work are the same skills that researchers use when they're designing experiments or they're analyzing data or they're coming up with their own explanation for the patterns that they're seeing in the lab. And so my way of integrating this into the classroom is giving you a creative assignment. And so essentially, the assignment is to create some kind of creative work that references something that we discussed in biochemistry this semester. It's due the last day of class, and I like to talk a little bit about their creative work. And it can be literally anything. I just ask that it's not a term paper because you're having plenty of chances to write this term while you're not writing a term paper. You're doing reflections, and then we'll be answering questions about a paper. And the details are on supply. So I provided a project that students did last term. So I had somebody do a Christmas card where they think they wrote out Merry Christmas and peptide, but peptide was Christmas light. It was pretty cool. Someone did an illustrated children's book about glycolysis. Although we haven't gotten to glycolysis yet, but it also probably was a very helpful study tool because it hasn't happened yet. I've had people do interpretive art about DNA, and I had someone write an opinion on the CRISPR-Cas in humans debate that happened actually right around the exam. And then one last thing I want to say that I thought was kind of funny is someone is, I think it was dating profiles for different biological macromolecules. So like this one's guanosine, and it's looking for a cytosine, and we had someone with glucose. So just, and then we had someone also make a video, about Splenda, which is pretty cool. But she would murder me if she found out that I couldn't translate it for you, so I won't. I'll respect your wishes. But literally, there's anything. So I showed an example last semester before I had an example that I made a DNA quilt. But literally, you can do anything as long as it's appropriate and as long as it's not a term paper. And I guess as long as you can turn it in somehow. So more details on the website and the Sakai, and I'm happy to answer any questions. Okay, now, so that's it in terms of grades. Again, come talk to me. And some of you got little, some of you I had suggested perhaps you might want to talk about. Yeah, I'm happy to talk to anyone about study strategies for the next exam. Now, for today, okay. Big surprise. The ditto has cut out. We'll try again. So you'll notice today that the setup for the groups is a little bit different. I don't have you guys with assigned roles per normal. So I think I explained why I used particular roles for the beginning of the semester. And now we're moving more towards a little more lecture heavy with interspersed problem solving. And at that point, I don't necessarily think it's appropriate to have those very formally assigned roles. I also don't see a reason to have a recorder, just personal. So what we're gonna do is each day, the instructor will be kind of randomly assigned, and then within your groups, you can pick the reporter, so the person who will talk to the class, and then the reflector, so the person who will fill out the sheet. I still want to have a reflector because I still want you to give me feedback on how the class is going. That's really helpful to me to help you. But so today the instructor is going to be the person whose birthday is closest to today. And then you can again pick the reflector and the recorder, or the reporter, not the recorder. And just a heads up, on Thursday, again, is the paper discussion. So I'm actually going to post the in-class discussion questions either tonight or tomorrow morning, so you can know what's going on going into class. But I ask that you don't actually work on the problems until we get into class. At the beginning of class, there will be time for general questions, and you can ask questions as you go along, but it's going to and there's going to be like 8 question categories. All the categories are going to have 2 to 4 parts where you're basically going to walk through each figure step by step. And it would be the approach that I would take if I was analyzing the paper. How many of you here have read a scientific paper before? Okay, so good portion of you, but I'd like to think of what we're going to do on Thursday as a guided tour. And that pre-assignment as well as this in-class assignment will count for credit. For accuracy. And with that in mind, I'm actually going to have the questions due beginning of class next Tuesday. So you can work with your groups outside of class time, just in case we don't get through everything during class. The nucleic acids quiz will be next Thursday. For the nucleic acid quiz, you're on the hook for the nucleic acid structures. So you need to be able to draw them as nucleobases, nucleotides, or nucleosides. You need to be able to draw them individually or linked in a strand. You should know the difference between an RNA and then ultimately you also should be able to draw in the hydrogen bonds between the different bases. This quiz rather than the enzyme quiz, but will be done in class over 15 minutes. Yes, Sydney. I would say I would practice that skill, yes. Because also, even if you can draw the 2 complementary strands, that's going to help cement those lower level drawing skills as well. And then lastly, would there be any interest, any problem session for next week? There would be? Okay, I'll send out a doodle poll or a Google poll, and we can get something on the calendar for next week. Okay, great. So we'll go ahead and get started. So again, as you'll notice today, it's going to be heavily lecture with problem solving. And I have done my best to incorporate, or have not have done my best, but I have incorporated places where we're going to problem solve. And this time, instead of me giving you some problems and you're going to figure it out, we're going to practice and build our way up to the types of skills. And what in my mine would be the highest level applications of the material that we're going to talk about during this lecture. And you'll see what I mean. I'm going to stop taking up valuable time by talking. Here are the learning objectives. Again, these are meant to help you as you study. From now on, they're going to be contained within the PowerPoint instead of in a separate folder. Okay, so in the pre-assignment, you guys had a pre-assignment even though this is primarily lecture-based because I wanted you to get practice with the vocabulary and also to clear out some of those lower level learning objectives. In that video, we talked about the 4 common classes of receptors, and we're going to dive into each of them in more detail, especially the first type of receptor today. So we have G protein coupled receptors, or GPCRs, which we're going to talk about in a whole lot of detail, receptor enzymes, gated ion channels, and nuclear receptors, which we'll talk about in slightly less detail. But GPCRs are going to be the one we're going to focus on. And why do we care about our so GPCRs? So you saw in the paper that the mu opioid receptor is an example of a G protein coupled receptor. And so you probably know a little bit about its structure already. All GPCRs have 7 transmembrane helices that make a barrel shape. This shape is conserved across all GPCRs. This figure also highlights an important part of something that often gets neglected when we draw pictures of GPCRs, and that's we have an extracellular portion and a cytosolic portion. And those portions are not contained in the transmembrane part, and they're generally relatively or the ramifications for if you're trying to structurally characterize this molecule. So in particular, the interior of a cytosolic portion is going to be interacting with the G protein, and it's going to get phosphorylated, which is going to affect signaling, as you'll learn about in the pre-materials for Thursday. And so, but then, okay, so we see the structure, but why do we care about GPCRs? Well, they're basically involved in everything. And most people in this class, or many of you in this class, are interested in either health careers or going on to grad school. And it turns out that 1/3 of all drugs in the market target GPCRs, but also GPCRs are mutated in 20% of so all surface receptors are involved in cell growth and development. So it makes sense that if they play a role in cell division, if they're mutated, you could see uncontrolled cell growth. So, but also for this class, we care about GPCRs because they're involved in the mechanisms of opioids as well, specifically the mu opioid receptor. So this is a rundown of all the different parts of a G protein coupled receptor. So on the left is the generic component, and on the right is the specific component for the pathway that we're going to use an example, which is epinephrine signaling. So epinephrine, also known as adrenaline. So this is going to be the pathway that's going to be triggered when you're really scared out of your mind. Dr. Alexander would use the example of if you're being chased by a bear. So our first messenger, the molecule that starts off the cascade is epinephrine, which binds to the beta-adrenergic receptor. The G protein, which binds to the beta-adrenergic receptor, acts as a transducer, but then binds to another surface enzyme, adenylyl cyclase, which is our effector. The effector is the molecule that produces the so the first messenger is on the outside of the cell. The second messenger is inside of the cell. And the second messenger is what's going to trigger the signaling cascade that's going to see the it's going to lead to the physiological effects that we have. So the second messenger, in this case cyclic AMP, will bind to protein kinase A, and protein kinase A is going to literally phosphorylate everything, and then that will eventually lead to increased metabolism, increased heart rate, and increased respiration. And this happens for the epinephrine signaling in our muscle, liver, and in our adipose tissue or fat cells. So a little bit about epinephrine. Again, it's adrenaline. The structure is right here. We've already gone through and identified, but just a bit of review and agonist. So the ligand is the native target for protein. So it's what the protein binds to normally in your body. But there are also other molecules that bind that aren't the native ligand. So agonists are molecules that bind and trigger the same signaling pathway. So in this case, the second molecule here is an example of an agonist, whereas an antagonist is a molecule that binds to the receptor but doesn't cause the signaling. It prevents the signaling from happening. And propranolol is an example of an antagonist. And so again, epinephrine increases heart rate. You can see how that could be bad for somebody who has like high blood pressure. So molecules that block this pathway can be used to treat angina, arrhythmia, and hypertension. So if you're curious, now you know. So we're going to take a more of a deep dive look into the beta-adrenergic pathway. There's a lot of things that are involved. So I'm going to start by giving you a brief walkthrough of how this pathway works. We're going to look in at some of the individual parts, You're going to draw it for yourself for practice, and then you're going to answer some questions about what goes wrong when you mess with one of the specific parts of this pathway. So, to start, the beta adrenergic pathway, again, an analogous pathway is one that we see with the mu opioid receptor. But what happens in the beta adrenergic pathway is epinephrine is going to bind to its receptor, which is the beta adrenergic receptor. Then, once the epinephrine binds, it's going to cause a conformational change in the receptor. Now you'll notice the cytoplasmic portion of the receptor is bound to the heterotrimeric G protein in green down here. When the receptor changes shape, it's going to bump into the G protein. And this is going to cause the G protein to also have a conformational change. When its conformation changes, it's going to kick out GDP, which is analogous to ADP, and instead GTP, or guanosine triphosphate, is going to hop into the G protein. Now, when the G protein is bound to GDP, it's inactive. But when it's bound to GTP, it's activated or active. And so when this G protein becomes activated, the G alpha subunit is like, okay, bye, or bye, Felicia, or whatever you young Generation Z people say these days. And the G alpha will leave the other two subunits. It has this little tail that hangs out, an isoprenyl tail that hangs out in the membrane. It's going to slide over down the membrane until it bumps into the effector, which is adenylyl cyclase. Adenylyl cyclase, like the adrenergic receptor, is membrane bound, but it's actually an enzyme. And what adenylyl cyclase does is it takes ATP and it cyclizes it and turns it into cyclic AMP or CAMP. And we'll actually see that reaction on the next slide or two. Now, what CAMP does is it goes and it activates protein kinase A, or PKA. And then once PKA is phosphorylated, it goes and it phosphorylates other proteins, which in turn phosphorylate other proteins. And that cascade of phosphorylation is what's going to cause the cellular response to epinephrine, so resulting in increased glucose metabolism and heart rate and respiration. But eventually, with time, all things must come to an end. So eventually, cyclic AMP is going to be decyclized by this enzyme here, which would cause the response to slow down. All right, so now we're going to start focusing in a little bit. So first, we're going to look at cyclic AMP. So what is the activity of adenylyl cyclase? So again, adenylyl cyclase, it's a membrane-bound guy here. It takes ATP and the 3' hydroxyl. It causes the 3' hydroxyl to make a nucleophilic attack on the alpha, beta, gamma phosphate of ATP. The two phosphates, the diphosphates, are going to leave as PPI, and then you get cyclic AMP, where you're connected at the 5' phosphate and the 3' hydroxyl. Again, this is why it's so important to memorize those different positions on the base. And so, however, with time, again, cyclic AMP is going to get degraded, and this enzyme can undo that and give you AMP instead. Now, I don't know about you guys, but I certainly have partaken Turns out this is how caffeine works. Caffeine actually prevents cyclic nucleotide phosphodiesterase from uncyclizing cyclic AMP. So if you keep your cyclic AMP levels higher for longer, it'll help you stay more alert. So now you guys know how caffeine works. Excellent. We've already learned so much today. And then taking a look at what our second messenger cyclic AMP does. How does it trigger that awake response? Well, it starts with PKA. So PKA, protein kinase A, is a dimer, a heterodimer. So it has two sets where you have a catalytic subunit and a regulatory subunit, and they're connected by a dimerization. Anybody remember another example of an enzyme? Yeah, you do. Okay, Paige. Pardon? Yeah, so so it is an allosteric protein, but does anybody remember a specific example of an allosteric enzyme we talked about? There was a question about it on the exam. Yes, ATCAs. So ATCAs also had a catalytic domain and a regulatory domain or subunit. And so what happens here is the regulatory subunit acts like a like a trigger lock on the catalytic subunit. And because when once cyclic AMP binds to the regulatory subunit, the catalytic subunit is able to pop off and then go start phosphorylating. So that's how this works. So when there's a critical mass of cyclic AMP, it can cause the regulatory subunit to dissociate from the catalytic subunit, and then the catalytic unit can do its job. So what exactly does pKa phosphorylate? I've told you that pKa triggers a cascade that leads to cell and respiration. If you don't believe me, here is a list of all the enzymes that pKa phosphorylates and what their downstream regulation job is. I'm not going to walk through it, but for those of you who are interested, here it is. Okay, so we alluded to this a little bit with pKa when we looked at that big figure, but also when we talked about what caffeine does. But we can't stay activated forever, right? If our heart rate was always increased, I mean, I feel like mine is, or if we were always storing all of or breaking down all of our glycogen and we couldn't store it, we'd be in real trouble. So our body has to develop ways to turn off the signal. And we're going to talk about several, and you guys picked one and talked about one, or you talked about one aspect of the pathway. Some of you talked about one of these, desensitization. But we can also have inhibitory ligands binding, which we're going to think about. The receptor can be desensitized or it can be taken out of the membrane and be recycled. We saw that CAMP could be degraded, and actually we can inhibit that from happening. But also kinases are enzymes that put phosphates on a molecule. Phosphatases are enzymes that take phosphates off of a molecule. So you can have phosphatases running around and undoing the work of pKa and its target kinases. That's also something that's going on. And all of these different methods are going on in the cell. But we're going to talk, but then to complete our discussion of how we turn off the signal, we're going to focus in on two more methods that actually involve the G proteins themselves and the receptor. And so I wanted to take a zoom in on a closer look on the life cycle of heterotrimeric G proteins. Because from what I've shown you so far, we've only looked at 1 through 3. When the pathway starts before epinephrine binds, the G protein has GDP bound, it's inactive. When the ligand binds the receptor makes a conformational change, the G protein makes a conformational change, and its GDP gets knocked out. It binds a GTP instead. Then the alpha is activated and it moves over until it bumps and activates adenylyl. If you were to believe the figure that I showed you at first, you may think that the G alpha protein stays active forever. It does not. It turns out we call but it turns out that this enzyme is actually an enzyme. It has a GTPase activity, which means eventually it's going to hydrolyze its GTP into GDP, become inactive again, and when it's inactive, it will go back and reassociate with its old friends G beta and G gamma. And so the cycle completes itself. Now, with time, this will just happen. There are other things in the cell that can help this process. So you can have other proteins that go and interact with the G alpha and speed this up, but with time, eventually, it's going to hydrolyze the GDP and then go back to where it started. But this is one thing that is happening. This is a constant cycle. Another thing that I wanted to go into a little more detail about, and this is because this is talked about in the paper. pre-video, is that eventually receptors are going to be desensitized due to the intervention of beta arrestins. So when epinephrine binds and the alpha subunit moves away, the signaling pathway can proceed like we said. But there's another fate that can happen. So the G beta and the G gamma subunits can also dissociate and then recruit a protein called beta RK, or I'm just going to call it BARK. It's going to recruit it back to the receptor, which is then going to phosphorylate the receptor at specific serine residues on that disordered cytoplasmic tail region. What happens when BARK phosphorylates the protein, phosphorylates the receptor? A protein called beta arrestin can come in, binds to this phosphate, and what beta arrestin does is it causes the receptor to be like taken out of the membrane in an endocytotic vessel, and then it just hangs out inside the cell for a while until it reverts back to original state, and eventually it will be delivered back to the membrane. So if you want a signal to stop, so I really want my heart rate to stop being activated, one of the ways the cell that can do this is to lower the effect of the signal by temporarily removing the numbers of receptors on the membrane. So that's one thing that can happen. Another thing, this is more of an FYI because it's going to come into play on Thursday, is when the beta arrestin binds, it can also cause alternate signaling depending on the receptor. And so for the mu opioid, we're going to see that this can cause interaction with the MAPK signaling pathway. But this is more just an FYI to see where this comes in, because I'm going to be I talk about this in the pre-video for lecture 4. But for now, as far as we're concerned, when beta arrestin binds to the receptor, it means the receptor is going to be recycled. Okay. Now, that was a lot of information. Hopefully, I didn't talk too fast, but we're going to begin our journey of analyzing the receptors. And so our first step of that is to synthesize everything that I've walked you through, and I want you to make your own drawing of the pathway. You can work with your group, or you can work by yourself, whichever you prefer. This takes 3 minutes, and you can use the pictures and then the list of the different parts to draw the pathway for yourself. And then I'm actually going to circulate through, and for those of you who didn't show me your assignment, this could be biology. It's all just a bunch of blocks. If you want to draw your receptor like a triangle, I don't care. Yeah, of course. Oh, oh, okay. Okay, I think it's a G as alpha, but the GI what? but I know that for this pathway, looks like a GS alpha. Yes, the G alpha. Um, yes. So for this one, it's GS. Oh wow. I knew that was. I knew that was the I want. Whatever. Yes. Yep. So the G okay, the GP five it activates alpha. Yes. Yes. So the GTP is activating the alpha subunit, and then the alpha subunit flies away. It's like I have a GTP, I'm activated, I'm secret to you, I'm leaving. And the GB and the G gamma handle all these other things too, but we're not worried about that. Yes. Yes. Yeah, the G as alpha activates animal cycles. So when they're together, they're yes. So when you have the heterotrimeric G protein, when all the subunits are together, it has to be bound to GDP. It's not active. Once it's activated, it's going to break up. It becomes, yes, activated or self-actualized or whatever you want to say. And it's like, I'm going off on my own. I don't need you. I'm going to go do some other things now. Okay. So if you're not quite finished up with your drawing, that's okay. You can work on it while you're working on the next couple of problems. But we're going to start now by beginning the process of one of the things that scientists are interested. One of the reasons why scientists are interested in G proteins is because they're implicated in so many diseases. And as we've learned from the first, especially the first exam, diseases often arise when something is not quite right with a protein. And if these pathways are involved with cell growth and cell division and our metabolism, if something goes wrong with one of the actors on that pathway, it can help us understand and then fix the problems that manifest themselves in diseases like cancer. So today we're going to see what happens when you get cholera. Okay, anybody here by any chance a fan of Victoria on PBS? No, I'm revealing myself as a crappity old person. Okay, that's fine. Okay, so cholera, really bad disease of the 1800s, transmitted through contaminated water, as they learned in Victoria. And what the cholera toxin does. So again, if we have our unactivated G protein here, if we have NAD and then the cholera toxin, what happens is then the combination of the cholera toxin and NAD can cause the ADB portion of NAD to become covalently bound to the G protein. And what happens is that ADP ribose looks a lot like ADP, but it's covalently attached to the molecule instead of just like dandy dandy hanging out in the pocket like GDP normally would. So what happens is the G protein is now permanently bound or semi-permanently bound to an ADP analog. And in this case, ADB would be analogous for GDP because they're both purines and they both have 2 phosphates, not 3. So let's think about what would happen to the consequences of the epinephrine signaling pathway if G alpha was locked into GDP. Okay, so if the G alpha was locked bound to GDP, where would it be located in the cell? Would it be hanging out with the other G proteins, or would it be moving over to activated adenylyl cyclase? Right. So if it's going to be the G alpha is going to be inactive. And so it's going to hang out with the other G proteins. And what this means is that it can't activate adenylyl cyclase. Now, if adenylyl cyclase can't be activated, what's going to happen to the CAMP concentration in the cell? Increase, decrease? Let's see a show of hands. Right. The cyclic AMP concentration is going to be decreased. And if the cyclic AMP concentration is protein kinase A going to be activated or deactivated? Pardon? It's going to be activated or is it activated or deactivated? It's going to be deactivated. So pKa is going to be deactivated because if we look back at the mechanism of pKa, when there's no CAMP bound, the regulatory subunit locks the catalytic subunit into not being able to do its job. And so if pKa is not activated, what's that going to mean in terms of metabolism? So glucose metabolism and then heart rate. Are those going to be increased or decreased if pKa can't phosphorylate things? We're going to see both being decreased because the activity of pKa phosphorylating other enzymes turns on those genes that make the proteins that cause our heart rate to go up and our glucose metabolism to go up. If we shut that down, our heart rate's going to go down and our glucose metabolism is going to go down as well. And so this is how if you change one protein on one pathway, you can see all these crazy things happen. And we're just considering one set of consequences, not the cross interpathway consequences, because that's way too big for us. Okay, so this is the kind of logical pathway that you're going to be taking when you're thinking about this pathway at a deeper level. So we're going to practice. I'm going to turn it back onto you guys now, and I'm going to give you 4 potential options. And I want you to work through this list. I want you to do which of these letters would have the same long-term effect as that ADP ribose analog from the cholera toxin. And so I'm going to start by giving you 2 minutes or a minute or 2 to work on your own, and then I'm going to have you report back to the group. So let's start. So let's go ahead and start working individually for a minute or 2, and then I'll have you report back. There can be multiple answers. Okay. So for those of you who've been working individually, take the time now to talk to your group mates. And then once you've come to a consensus, send up the person that you've designated to be your reporter to come write your answer on the board. Yep, go for it. That's so embarrassing. No, no, no. It's all good. Hmm. Okay, guys, I have to admit to something a little bit embarrassing. Okay, so I actually, when I was preparing my lecture, I actually misread my notes, and it turns out that ADP ribose actually locks it into an active state, not an inactive state. So what we're gonna do is what we're gonna do with our notes here, and again, I sincerely apologize for confusing this. I keep betraying myself as a protein structure biochemist here. What we're gonna do is when you're going back to your notes, just cross this out, and we're going to assume that it's just always bound to ADP. So we're going to disassociate this situation from the cholera toxin and just say, let's pretend we're in a situation where the G protein acts like it's always bound to GDP instead of GTP. I know it's confusing, and I sincerely apologize. The actual toxin causes it to look like it's GTP, but we're not going to worry about that. We're just going to assume that we're locked into a GDP bound. Yes, yes, Kendall. Pardon? Yeah, we're going to act for this discussion. The cholera toxin causes it to act like it's bound to GTP, not GDP. So know that the cholera toxin causes it to be active all the time, but for the sake of our discussion, we're just going to, oh goodness, how I pulled myself out of this. Okay, but for the sake of this discussion, where we're saying what happens, we're gonna forget about the cholera toxin, and we're just going to say what happens if the G protein acts like it's bound to GDP rather than GTP. So that way we don't have to go and do this thing where I told you the wrong thing about cholera. We have to relearn it and change the whole thing. We're just going to say what happens if the receptor acts like it's bound to GDP, and know that there are situations where the receptor could act like it's always bound to GTP. Does that make sense? Are we on the same page? Okay. So proceeding forward, again, I sincerely apologize. A little embarrassed here. That's okay. Mistakes happen. We move on. If we have a problem where the protein acts like it was always bound to GDP. Okay, and it looks like most of our groups had some variation on B and C and D. And so A wouldn't act like the same thing, because if you had a situation where the G protein was constitutively active, so constitutively active being always on or always activated, so it was always bound to GTP, you would actually see the opposite effect. But all of these mutations are going to cause a lower concentration of cyclic AMP, and so they would cause the downstream effects to be turned off as well. So whether GTP can't bind the receptor, GTP can't bind adenylyl cyclase, or if the G protein can't get rid of GDP, all of these are going to contribute to adenylyl cyclase not being activated, cyclic AMP not being produced, PKA not being activated, and then metabolism and heart rate not being dialed up. Yes, Hubert. For information, yes, but I'm not going to hold you accountable for the cholera toxin as an example, considering I messed it up. That wouldn't be said that I wouldn't say what would happen if the receptor was always bound to GTP, but I'm not going to put you on the hook for specifically the cholera toxin if I told you the wrong thing in class initially. That makes sense. If you're confused, still confused, come talk to me after class and we will straighten it out. Okay, so now what I want you to do is I want you to consider an alternate reality here. Let's say, and actually this is an alternate reality that's real. Let's say that the receptor interacted with the G protein that turned off the what would happen to the AMP or cyclic AMP concentrations, the PKA activation, and then the heart rate? I want you to take a minute and work with your groups to record what would happen to the cyclic AMP, the PKA, and heart rate if the G protein deactivated adenylyl cyclase. Go ahead and work on that now. Okay, so let's report back. Okay, so if we have a G protein, so if the receptor interacts with a protein such as the GI, or the inhibitory protein that then turns off adenylyl cyclase, what would happen to the cyclic AMP concentration? Would it increase or decrease or stay the same? Right, so cyclic AMP concentration is going to go down if the protein turns off or does not activate adenylyl cyclase. How about PKA? Will PKA be activated or deactivated? PKA is going to be deactivated. And what about our heart rate? Is our heart rate ultimately going to go up or going to go down? Down, right? So again, this is the type of logical path, this is the type of thinking that I'm going to want you to take as you're working your way through these pathways. And some more questions or to think about in this class, but you can also see how if you activate things more than normal, you could see an increase as well. And so these are just some steps to think about as you're going through and studying these pathways. One last topic on G protein coupled receptors I wanted to talk about is the idea of amplification. So we saw an example of one epinephrine molecule binding to one receptor, but it's not a 1-to-1 ratio. So for every one epinephrine molecule that binds one receptor, you have 10 GS alpha subunits activated. And as you go down through the process, so we end up with 200 molecules of cyclic AMP, 100 molecules of active pKa, and so on and so forth until you get to the end, which is increased blood glucose, and you end up with actually 100,000 molecules of glucose for a single molecule. So you have like a 5 or 6 order of magnitude increase between your first messenger and your end product. So the amplification is really very powerful. Yes, great. When it's activated, it can have other G proteins come up to it and then be activated and leave. Yes. I actually don't know. I would think it would depend on the pathway in the particular molecule. I think it would depend on whether or not the beta and the gamma leave. Okay. All right. So we're gonna breeze through the other types of receptors, which are also important, but we didn't spend quite as much time on because perhaps they're a little bit less complicated in terms of moving parts, but also because we're talking about a GPCR on Thursday. So an example, the way ligand-gated ion channels work, and then a specific example is the acetylcholine receptor, is these ligand-gated ion channels they're a bundle of alpha helices that hang out in the membrane. And they're amphipathic helices, which means they have a hydrophobic side and a hydrophilic side. So when the receptor is not activated, it has its hydrophobic side pointed inwards. And when the hydrophobic sides are in, it has like leucines that are kind of bulky, so it blocks the channel so nothing can pass through. But when acetylcholine binds to the receptor, it causes those helices to turn and then twist out, and then you have hydrophilic or polar amino acids lining the inside of the channel instead. So you can literally visualize the helices rotating and changing their face. And so when these helices rotate and you have the polar amino acids on the inside, you can have ions flowing in and out of the cell. Now, why would we care about this? So this is one of the ways that neurons state one of the ways that helps neurons signal to each other in the cell. So the cell has a membrane potential of around -50 to -70, so an average of -60 millivolts. And what this means is that the inside of the membrane is negatively charged, the inside of the cell, the outside of the membrane is slightly positively charged, and the difference is slightly in favor of the negatively charged inside. Now, let's say we have a neuron. There's some kind of signal that causes this voltage-gated ion channel to send sodium into the cell. So the inside of the cell is slightly negatively charged. So if you have positive charges going in, it's going to depolarize that membrane potential. So a cell is going to compensate by repolarizing the cell by kicking other ions out. And so this process of the depolarization and then the repolarization of the cell is going to cause secretory vessels that have acetylcholine in them to be released. And so the cell, in response to this polarization, depolarization, and repolarization, is going to make vessels with acetylcholine. Those are going to go to the membrane surface, and in the opposite of what we saw with receptor cycling, the acetylcholine will then be released into the space between the two cells, or the synaptic cleft. Apologies to you neurochemists if I am butchering this. And the acetylcholine is going to go to the surface of the next cell, and it's going to bind to the acetylcholine receptor. And so when the acetylcholine receptor binds, before it's not letting anything in, but now it opens up, it's polar, and so calcium and sodium ions can flow through, and that's going to trigger the next wave of depolarization and repolarization. And so this is one way that signaling could be facilitated between neural cells. Okay, the next set of receptors are receptor enzymes. So and ligand-gated ion channels they have molecules bind to them that cause a conformational change. But the receptors aren't catalyzing a chemical reaction. They're just undergoing a conformational change. Receptor enzymes are actually themselves enzymes, and they themselves catalyze a chemical reaction. So one example of this is the insulin receptor. And what these receptors do is they autophosphorylate or they phosphorylate themselves. So with the insulin receptor, again, it looks a little bit different than the other receptors we've seen. It has little transmembrane helices, but most of it is actually extracellular and intracellular. But when insulin binds to the extracellular portion of this receptor, it undergoes a conformational change, just like we saw with all the other receptors. Except this time, the intracellular portion of the receptor is going to phosphorylate itself, or autophosphorylate. And so once this receptor enzyme is phosphorylated, other proteins are going to bump up against the receptor and then become phosphorylated. And this will lead to a signaling cascade that is similar to what we saw with PKA and the epinephrine signaling pathway. Now, insulin signaling is really important. Already, if you hear about insulin, I know it makes me think about diabetes for one. It turns out that insulin has its finger in several different metabolic phenotypes. So one is that insulin is actively involved in cell division. Now, I'm not so concerned about you knowing all the individual portions here. What I want you to know is that the activation of the insulin receptor by the binding of insulin eventually leads to transcription or expression of proteins that help with cell division. So one of the downstream results of the activation of the insulin receptor is that cells are going to grow and divide. That's one. The other big long-term effect of insulin signaling, and this one perhaps is a little bit more obvious, is that insulin binding stimulates glucose uptake and storage. So when the insulin receptor is turned on, one of the things that happens is that the enzyme that breaks down, so one of the things that happens is that glycogen production from individual glucose molecules is turned on. And the specifics of that pathway are here, but one of the things that insulin signaling causes is for glucose being turned into glucose to be turned into glycogen. So if the insulin pathway is turned on, that receptor is activated, it downstream leads to increased cell division and increased glucose storage into glycogen. Yes. Pardon? Yes, it has to happen in response to insulin binding. Yeah, yeah, that is a direct response. So the GPCR, the epinephrine, when the epinephrine binds, the receptor changes place, and then it causes a conformational change in the G protein, which kicks off the signaling. In this case, the triggering event after insulin binding is the receptor phosphorylates itself. Okay, so we're going to take another minute to practice a quick skill. We'll allow like maybe a minute or two. So let's say we have the insulin receptor. The insulin receptor has an amino acid, a glycine, that is turned into a valine. This is purely hypothetical. I made this up. So let's say there's an amino acid on the insulin receptor, and this mutation does not allow insulin to bind to the receptor. What would be the effect on cell growth and then glycogen synthesis? So we'll say glycogen synthesis. So if insulin can't bind to the receptor, how would cell growth be affected? So cell division, and how would glycogen synthesis? Okay, take a minute or two and walk through that. And when you're ready, why don't you send a new recorder, and then just draw arrows in the up or down direction for each. So the first arrow will be cell division, and the second arrow will be glycogen. All right. So looks like we've come to consensus on the fact that if insulin can't bind the receptor, we would see a decrease in cell division, and glycogen storage would be decreased as well. So we perhaps would see an increase in the individual glucose concentration in our blood. So it's much, much more complicated than that for like diabetes. But you could see how this pathway being messed up could affect the level of glucose in our blood sugar, as well as whether or not our cells grow and divide. There's also another situation here, but I'll let you work through that on your own time. Now, receptor enzymes there are many more receptor enzymes out there. One class of receptor enzymes are growth factors. And one particular one shown here is the epidermal growth factor. So epidermal means it's on your skin surface. And so oftentimes these receptor enzymes are tyrosine kinases, so they autophosphorylate at tyrosine tyrosine residues. And so mutations, if these are tied to cell mutated, so they're always on, so they're always triggering cell growth and division, how that could lead to cancerous cell growth. So again, these also are potential therapeutic targets, these receptor enzymes. So the last class of receptor are nuclear or steroid receptors. And these nuclear receptors are different from the other three receptors, which are surface receptors, because steroids, as you may or may know not from previous chemistry courses, they're hydrophobic. They're usually planar. And this allows them to pass through the cell membrane without the aid of a receptor. And so what these guys do is they pass through the cell membrane and they go into the nucleus. And then once they go to the nucleus, they bind to a transcription factor. And this transcription factor then turns on the transcription of a certain set of genes for proteins that are involved in a particular process that wants to be upregulated. And so the receptor for these molecules are actually in the nucleus rather than on the cell surface. Now, these receptors generally have a slower effect than the surface receptors because whereas the other receptors communicate through phosphorylation and allosteric changes, this actually has to turn on the production of a protein to see the result. So this receptor, even though in terms of perhaps structure it's a little bit simpler, the effect is going to take longer to kick in because you have to make your proteins instead of just like phosphorylating. Okay, and so one more activity that I thought of for this one, I don't know if you guys are familiar. Here are some molecules that are involved in cell signaling. So why don't you guys look at your table and venture a guess and circle which one of these that you think could act as a nuclear receptor. Yes. I I was gonna say, okay, maybe so. Okay, so are there any groups in particular that would like to volunteer? Which one they came up for? Which ones could act as steroid receptors? Yes, Elena. The top right. Okay, why would you think the top right? Okay, so one thing that, okay, so what I'm gonna do is I'm gonna actually put up a picture of cholesterol, which is a molecule that hangs out in cell membranes. And then I want you guys to look at this question again. Cholesterol picture. Oh, Jay Leno. Oh, I'm so glad I knew that. Okay, here's this picture of cholesterol. So cholesterol is a molecule that hangs out in cell membranes. So looking at the structure of cholesterol, I'm glad you guys took a guess, and I appreciate the logic of associating the hydrophobicity with what would work. But here's a molecule that's known to hang out in cell membranes. So looking at the structure of cholesterol, there's your answer. But I don't know that for certain. Okay, so if we look back at the front, steroid, most of the molecules that act as nuclear receptors are steroids, and a characteristic of steroids is that they have a fused ring structure. And so if we look at these molecules here, this is cortisol. Whoops, okay, of course I don't have it. We have cortisol and estrogen. I think estrogen. So these two are molecules. They have a characteristic fused ring structure. I'm not sure why physiologically, but there's something about the four fused ring structure as probably, maybe it's perhaps it's approximately the size or can wedge in between the tails. This allows these molecules to pass through. There are some hydrophilic groups on these molecules, but overall the dominating characteristic is going to be these hydrophobic rings. These molecules, so we have serotonin, I think that's limonene over on the top right. These are molecules that are actually ligands for GPCRs. They just happen to be hydrophobic. And so the difference is going to be that four fused ring structure. And we're not going to talk about this, but if you're interested, when I taught this class last, right about this time, there was the whole brouhaha over LaCroix not being natural or whatever, and people were and there were lawyers that were alleging that limonene and linalool cause cancer. And so I made this little slide to alleviate the fact these are in fact natural compounds. They may be synthesized in a lab, but they actually play a really important role in our cycle. So just FYI. And I know it's about time and you guys are itching to leave, but so for this slide, what I would say that you need to know about G proteins is that G proteins can be manipulated. So either they look like they're always bound to GP, or they look like they might always be bound to GP. And you should be able to analyze the effects on the signaling pathway if that happens. I'm not going to specifically hold you accountable for the cholera toxin because I don't want you to get confused. And again, I apologize for my momentary lapse there, and thank you for the kind Samaritan who pointed that out to me. Again, I'm human, and so if I make a mistake, please tell me. I'll try to minimize my mistakes. And it's 10:30 now, so I'm gonna let you guys go. Be sure to answer the pre-assignment, read the paper, look through the supporting information, and if you get a chance, look over the in-class discussion. And we're gonna talk about some opioid receptors on Thursday. Oh, that was a scary sound.